Welcome back, friends. Welcome back in another molecular marker video from microbiology with Shaknik. So in this video we will going to discuss about random amplified polymorphic DNA or RAPD. Okay, in AFLP I have promised you to discuss about RAPD. So today's video is for RAPD. Before going to discuss this video, I want to apologize for making a mistake in AFLP that I will going to resolve here. That in AFLP I have told you that RFLP is a dominant marker, right? But it is not. RFLP is a co-dominant marker. Okay, very sorry for that. That means codominant marker means we have discussed in the very first video of molecular markers that codominant markers have a special capability of distinguishing between homozygous and heterozygous, right? That means it can tell you whether your DNA sequence is coming from a locus which is homozygous or heterozygous, right? Along with it have the ability to distinguish between dominant or recessive allele also. Okay, so today's discussion, that is RAPD, is the dominant marker. Okay, this one is the dominant marker. Okay, so now let's talk about the introduction. So RAPD is a dominant type of molecular marker. Dominant type of molecular marker means here it have the ability to distinguish between. Okay, that means this line will be it have the ability to distinguish between. Okay, again sorry for this. So now RAPD is a dominant type of molecular marker. So I hope you know about dominant type of molecular marker. If you don't know, let me explain in short, and if you want to understand it in details, then please go and watch my first video on molecular marker. So dominant type of molecular marker means this type of markers can help you to distinguish between dominant allele and recessive allele. Okay, and it can also distinguish between polymorphic DNA and monomorphic DNA. Now the question is, what is monomorphic DNA and what is polymorphic DNA? So let me tell you that in very first video I have told you that in between me and you there is 99.9% similarity. Okay, and this 99.9% similarity is because of the monomorphic DNA. That means this monomorphic DNA contain same DNA sequence at particular location of the chromosome in you and me. Okay, and in among every individual in our universe. Okay, and polymorphic DNA means the 0.1% which is responsible for the difference between you and me. Okay, and also have the ability to distinguish between dominant and recessive allele. So but it cannot distinguish between homozygous and heterozygous genotype. Okay, homozygous means homozygous dominant. Mind it very carefully. Okay, you may heard about that it cannot have the ability to distinguish between homozygous and heterozygous genotype, but the answer is unclear because it have the ability to distinguish between homozygous recessive and heterozygous. Okay, but it cannot have the ability to distinguish between homozygous dominant. Okay, so I hope it is clear now. Now let's try to focus on the full form of the RAPD, that is random amplified polymorphic DNA. What it wants to tell us? It wants to tell us that the primers here we are using, because it is clear that as the amplification term is present here, that means we will going to use the PCR, right? That means it is a invention of post PCR era, right? And another important thing, just focused on the first word of the total random amplified polymorphic DNA, that is random. Okay, random means here the DNA is amplifying randomly. But is this a feature of normal PCR? No, because in normal PCR we are using specific primers that will ensure the selective amplification of the DNA, right? So that means here we will use the primers which will be non-specific. Okay, that is a very important feature of the random amplified polymorphic DNA. Okay, so remember it very carefully that here we are using non-specific primers which are nothing but short single stranded nucleotide of 8 to 12 nucleotide long. Okay, so what we are doing, so first of all we will isolate the sample. Okay, and then we will isolate the DNA, then we will use PCR. That PCR involves non-specific primers, and then agarose gel electrophoresis, and then the profiles, that RFID profiles that we have get, we will compare, right, so that we can get the result. Okay, now it is important to understand about the PCR step in case of RAPD. Okay, more specifically the step where annealing of primer with the template DNA is going on. So let's see. So first of all, the denaturation step where the double stranded DNA will be separated into two single stranded DNA, right? Then at 55 degrees centigrade annealing will occur, right? Now during this annealing there can be two possibilities. Okay, one possibility is, let me draw it, then it will be very easy for you for understand. That suppose, okay, sorry, it is a double stranded DNA molecule. Okay, so we have denatured it, then we have added the primer. Okay, so now let's say that, I hope you know that PCR involve two primers, right? Forward primers and reverse primer. So let's say first two forward and reverse primer attached in such a way that I have mentioned here. Okay, and another two types that is attached in, let's say, okay, let's say here and here. Okay, so now in case of the first one, amplification will occur. Okay, because as you can see that the forward primer and the reverse primer are closely attached, right? But here there is a significant difference between the forward primer and reverse primer. Okay, under such circumstances there can be a chance of that the amplification or the extension will not occur. Okay, especially when the difference between two primer is more than 2 kb. Okay, so as a result, as you can see that various kind of primers are attached at various places. That means it will encourage the formation of random length DNA, right? That means the PCR product that we will have will consist of random length DNA. Okay, that length will be varies, but try to understand that when we are using the normal PCR technique, you cannot get such type of thing, right? Because there you will use the specific primers, right? And as a result you will get the PCR products which are of same length, right? But here you cannot get this. Why? I think you can understand. Okay, now we will run them in agarose gel. Okay, and then we will can analyze the genetic variation of an individual. Okay, now it will be important to understand the application of the random amplified polymorphic DNA in real life. Okay, so again we will going to take an example of criminal case. Okay, so let's say you are a detective and you have isolated a let's say hair sample, okay, from a crime scene. Okay, now your goal will be to transfer it to forensic scientists. Okay, now those forensic scientists will isolate the DNA from here. Okay, now those DNA will be used to perform the random amplified polymorphic DNA. Okay, molecular marker test. Now, let's say as a detective, you are suspecting two individuals, suspect 1 and suspect 2. Now, you are also apply the same technique for suspect 1 and suspect 2. Okay, now you have get, let's say, so what you will obtain? You will obtain 3 agarose gel plate. Okay, the first one is for suspect 1, second one, let's say, the agarose gel of crime scene, and the third one is suspect 2. Now your goal will be to compare the 3 agarose gel and find out in which case which two plate contain same DNA bands. Okay, again I am repeating, your job will be to, or your forensic scientist will find out the similarities between the 3 agarose gel plate that you have. Okay, so as you can see that you are getting similarity with suspect 1 and crime scene. Okay, that means what? That means the suspect 1 may be present during the time of the crime. Okay, at that scene. So that's how the random amplified polymorphic DNA are using. That is how we are using random amplified polymorphic DNA for forensic sciences. Okay. So if we try to understand it again, that we are taking a sample from crime scene, and now from that crime scene, so you have obtained a sample from the crime scene. Now you have isolated the DNA from the sample. Now you have applied the random amplified polymorphic DNA. Now as a detective, you are suspecting two suspects. Now you have also ordered them to go and give their sample to your forensic scientist. Okay, so they have given their samples. Okay, from that samples your forensic scientist isolated the DNA, and now from that DNA he have performed the RAPD. Okay, and after the test you have get, or your forensic scientist have get the 3 agarose gel plates. Okay, now he or she will compare the 3 plate and try to find out that which of the 2 plate are resembles. Okay, or which of the 2 plate are similar, right? So as you can see that the suspect 1's agarose gel plate and the crime scene's agarose gel are quite similar, right? So we can conclude that suspect 1 may be your criminal, right? So now application. So forensic studies that we have just discussed, along with gene mapping studies, cross in variety identification, microbial strain determination, we are doing with the help of this RAPD. Advantages: the most important advantages is that unlike PCR, we don't have to know any important sequence information of our DNA, right? That we have to when we are using PCR. Why? Because in PCR we are using specific primers, right? And with the help of the random primers, we can analyze the genetic diversity of individuals, and it can be done in low amount of DNA, and that is obvious, right? Because the step named, or the process named PCR is involved here, so we don't need to bother about the amount of the DNA. Okay, because we can amplify it. Disadvantage: low reproducibility, and another important thing that I have already told in the introductory part, that our RAPD marker is the dominant marker, right? That means you can distinguish between dominant and recessive allele very well. Okay, and it can also give you knowledge about the difference between polymorphic DNA and monomorphic DNA, no doubt about this, but it cannot give you information whether your DNA sequence is being amplified from a locus that is homozygous or heterozygous. Homozygous means homozygous dominant or heterozygous. Okay, we have discussed about this in the very first molecular marker video, that there I have told you that two biological parents, okay, claiming a biological claiming a children or claiming a child as their biological child. Okay, and as it is under such circumstances, if you use dominant markers, then these dominant markers will either give you similarity of the children with father, either mother, right? Let's say the black band that I am drawing here is the DNA from mother, okay, and this green is both the cases is the children. Okay, now suppose it is agarose gel 1, agarose gel 2, you can get either one result. Okay, that means either the gel plate will showing you that the DNA of the child is similar to the father's DNA, and either the gel plate will show you that DNA of the child is similar to the mother's DNA. Okay, under such circumstances, if we consider that the DNA from the father's one is dominant and mother's one is recessive, then with the help of the result you can either conclude whether the band is from dominant or recessive, but you cannot conclude that whether the genetic makeup of the children is heterozygous or homozygous dominant. Okay, so that is the problem of dominant marker, and that was solved by codominant markers such as, for example, RFLP. Okay, so now another one, that here as here we are using non-specific primers, there can be a high chance of mismatch between the primers and the template, and as a result, in most of the cases you may get very low amount of product, or in some cases you don't get any kind of product. Okay, and as under such circumstances the result become very difficult to interpret. Okay, so now as we are here in the era of high throughput sequencing, in this era those kind of techniques, let's say for example RAPD or RFLP, are no longer too much relevant in these years, right? But in 90s RAPD was the technique that we are using. Okay, and another important thing you have to remember that it is a post PCR era invention, right? Because here we are using PCR, right? And we have started our journey of molecular markers from RFLP, right? Random fragment length polymorphism, right? Here we are not using PCR because it was the pre-PCR era invention. Okay, and AFLP is nothing but the mixture of RFLP and RAPD, another post era invention, post era PCR invention. Okay, so this was all about the discussion of RAPD. So it was all started from the full form of the RAPD. So as it is containing a term called random, that means here we are using PCR, but here we are using also non-specific primers, right? That causes amplification of DNA randomly, right? And then by using agarose gel we can get band patterns, and by comparing band patterns we can consult about the genetic variation between individuals, right? And again, important thing I want to mention that it is a dominant marker. In previous video we have discussed about RFLP, that was the codominant marker. Okay, so that's it for today, friends. So I hope this video will be helpful to you to understand the basic concept of the random amplified polymorphic DNA. So till then, we will again going to discuss about another kind of molecular marker that I want to discuss, that is SSR. So till then friends, bye bye and take care.